Recording. All right. So we have an exam next Tuesday. I have, as of last night, I completed your practice exam. Um, if you're thinking, how should I study for this exam? I can just tell you that it's a very good use of your time to take a look at that um, to take a look at that practice exam, work through that practice exam so that you basically know each question, why it's right, why it's wrong. Um, and more than that, for each of the questions that you see on the practice exam, I would take those and say, okay, if I change this element of it, am I changing the question? For example, one of the things that we went over in this block of material is we talked about, pulling up my PowerPoints here. Sorry about that, everybody. We talked about sugars. Now I'm going to just kind of walk through all of the different PowerPoints. And okay, so in addition to that, I wanted to walk through kind of the grading. Okay, first things first, what I wanted before I get into the content, I just kind of want to give everyone a refresher about basically how this class is graded. Um, and in the event that you're good on this, then don't worry about it. So I think that this is always a helpful refresher and a reminder. Um, so this is kind of the rundown of what I see on my Canvas site. It's a little bit different from what you see on your Canvas site. Um, each of our 5 in-class exams is worth 12% of our grade, or 12% of your grade. And then your final exam is worth a total of 15% of your grade. So it's a little bit more. Um, if you look at exam number 1 here, or what you see here is exam number 1, you will see that there are 3 items. Now, exam number 1, you took 1 exam for that, and that is indicated here by remotely proctored, this one right here. Now, that was taken on September 17th. Then on October 8th, you took another exam. And let's say that on exam number 1, you got a 80 on that. Then on exam number 2, the second time that you had an exam, you got a 100 on that. Well, what I'm going to do is I'm going to take this grade and write it here as well. So that means you get a 100. Okay. Now, this third item down here, exam number 1, take 3, that's the score that you're going to get on your final exam. Let's say that on your final exam, you get a 95. Well, any place in the grade book where you see a take 3 final exam grade, you're going to see that 95. Now, what that means is that these 3 grades are all in consideration for that 12% that reflects exam number 1. But I want to bring your attention to this right here. Rule number 1, one rule. That one rule says that, or I set that rule to drop the 2 lowest grades. So if on exam number 2, you got a 100, on exam number 1, you got an 80, and then on the final, you get a 95, only the 100 is going to be used for your grade calculation. So effectively, what that means is that this 80 and this 95 are dropped from this calculation right here. Now, this upcoming Tuesday, you're going to have exam number 3, and that's going to be a remotely proctored exam using Proctorio. And let's say that on that you get I'll just label this as exam 3, take 1. Let's say that on that you get let's say you get a 75. Well, that 75 is going to be populated up here for your exam 2 calculation. And then your final exam grade, whenever you take that, that's going to be a 95. Exam number 4, let's say you get a 70 on that. Okay, so now what we're talking about is calculating your exam 3 grade. Well, you're going to have the same rule, drop the 2 lowest. And so that's going to drop the 70 and the 75, in which case having done really well in the final exam is to your benefit and is a great thing. Okay, so effectively what that means is that on Tuesday, when you take your exam, you have the opportunity to raise up your exam two score. And then if again you don't do as well as you want to on this upcoming exam on Tuesday, the final exam is your last chance to really boost your grade altogether. Now, if you're thinking to yourself, "Oh my gosh, this is kind of a great opportunity," I don't disagree with you. Um, if you're thinking to yourself like, "Oh my gosh, does this guy like I could skip all of the all of the exams, all the midterm exams, exams 1 through 5, and then if I just ace the final, man, that puts me in a great position." Yes, you're absolutely right. Because the day that you take the final, you've had the opportunity to see everything, reflect on everything, um, you know, review the right answers on the exam questions. You got that chance, and that's a really great chance to bring your grade up. So if you're thinking that, you're absolutely right. Do I think that's a really good strategy to skip every exam and then take the final? I don't. Um, have I done this before? Yes. Has anyone skipped all of the exams and taken the final? Yes. What happened to that person? I think that person, this is in an engineering chemistry class, I think that person got a 26 on the final, and that was a 26 out of 100. I don't know if it was their strategy to do that, to just like boost everything up. But if that's what their strategy was, it didn't work. It didn't work in their favor. So I just caution anyone and everyone about that. But please take this as an opportunity to bring your grades up. Exam three, great. That can totally offset exam number two. Um, at any rate, now I'm going to go back to sharing my screen. We're going to talk about sugars. Um, the practice exam that I gave you guys, that I posted on our Canvas page, reflects the material on the actual exam that you're going to take on Tuesday. So what that means is basically your first roughly 8, maybe 9 questions are questions over the exam 2 content. So if you look at our Canvas page, what that will show you is that the exam 2 content was, I'm pulling it up so I can use it as a reference. It's not about drug design, it's instead about amino acids, single letter codes, 3-letter codes, um, protein structure, the different levels of protein structure, as well as a little bit of protein function. So with that in mind, um, we are going to talk about, oop, went too far. We are going to talk about sugars. So when it comes to sugars, there's two major molecules or two major groups. Those are your aldoses and ketoses. Now, that should sound a little bit familiar because aldoses and ketose, aldehydes and ketones were functional groups or organic functional groups. Now, this right here is an example of an aldehyde. So where I put the check mark, there's actually a carbon. What that looks like is that carbon, or another way to look at this molecule here is C. So that's one way that you can look at that molecule. That's the same, this molecule that I just drew is the same as this molecule right here. Now, lots of questions that I have about um, sugars are basically classification. So when you look at this molecule, I want you to be able to say, answer a question about this molecule. Um, For instance, one thing that is true about this molecule that I circled is it has 6 carbons, meaning that it is a hexose. If a carbon has 5 carbons, it is a pentose. Um, what is a 4-carbon sugar? Add to the chat what a 4-carbon sugar is also known as. give you a hint. It ends with os, but what is the prefix? What comes before os? Perfect. Perfect. It's a tetrose. Three carbons would be a triose. So if I ask you a question where I show one of these sugars and I ask you, what is this? Well, one thing that it is a, D3-ose here. I'm not going to ask you for the common names of these sugars, except for this one right here, glucose. That is a tetrose, or D3-ose is a tetrose. Now, I also will ask about a ketose. So I'm going to clear all my drawings here. What this shows us is basically our differences between two sugars. We have an aldose and a ketose. Now, what I do, the sugar that I most want you to know the structure of is glucose. Why? Because it's relevant to almost everything. Glucose is it's a major energy source for any organ or for most organisms, except for plants. Plants make it. So I want you to know the structure of glucose. In addition to that, I want you to be able to identify hexoses, aldoses, tetroses, pentoses, and trioses. So basically, how many carbons does a sugar have? If we look at these sugars right here, these are cyclic sugars. These are both cyclic forms of glucose. And what differentiates these two is the positioning of this hydroxide group. I'm going to put a check mark by the hydroxide group that I'm talking about, that one and that one. One of them's pointing up, one of them's pointing down. If it's pointing up, then it is a beta glucose, or it's the beta form of this sugar. If that hydroxide is pointing down, it is the alpha form of this sugar. So in addition to that, there are what are known as disaccharides. Disaccharides are sugars that are made up of two individual sugars. So sucrose is specifically a polymer or a dimer of glucose and fructose. Lactose is glucose and galactose. Maltose is glucose and glucose. I want you to know that. I want you to be able to kind of put that together. Then we've got, after this, we talked about nucleic acids. Now, our conversation around nucleic acids, I don't know why my file's not opening. Um, when it comes to nucleic acids, we talked about a couple of different things. We had DNA or deoxyribonucleic acid and RNA, ribonucleic acid. We talked about three different major kind of genetic uh, discoveries or experiments. Here, what we have is what's known as the Hershey-Chase experiment. And your takeaway for this is that this experiment concluded that DNA is the carrier of genetic information, as opposed to proteins. So around this time, people were really unsure about that. Now it's pretty well accepted that the Hershey-Chase, or sorry, it's pretty well accepted that your DNA is basically your genome, and that is what carries your genetic information. In this model or on this page, we talked about the DNA double helix and figuring out that structure. And it was really the work of four people. There was Maurice Wilkins, Francis Crick, James Watson, and then most importantly is Rosalind Franklin. Rosalind Franklin did this work right here. She kind of solved this like top-down view of a DNA molecule. She was like, this is what it looks like. And then people were like, ooh, I think that's a good idea. Ultimately, it was Maurice Wilkins, James Watson, and Francis Crick that were awarded the Nobel Prize for this, but they couldn't have done it without Rosalind Franklin's work. Then our last and maybe most significant and recent discovery was the work that was done by, or as a part of the Human Genome Project. This was a project that was taken on by the Department of Energy, the United States Department of Energy, the agency, as well as this fellow who's pictured here, J. Craig Ventnor. This was ultimately concluded in 2003 there were some benchmarks before then, like in the early 90s, there were other organisms that had their genome sequenced, but the human genome really, its completion was in 2003. Now, all that to say, what we're looking at is a molecule of DNA. There's the primary structure, which is the DNA sequence. This is read in a 5 prime to 3 prime direction, which might sound like, what is this 5 prime? Well, that's coming from this. Nope, that's not what I'm looking for. That's what I'm looking for right there. This right here, the 5 prime end of this sugar. So this sugar, 5 prime to the 3 prime end. So we look at the sequence A, T, G. That gives us our sequence of a primary or the primary structure or the sequence of a DNA molecule. Now, when we look back at this shows us basically the building blocks for a DNA molecule. One very important thing is that ribose, ribose is the sugar that is involved in both DNA and RNA. The only difference between DNA and RNA is that in a DNA molecule, ribose has been deoxygenated. So it loses this right here, this 2' OH group. Now that 2' is just indicating the carbon number in that sugar. So carbon number 2 in the sugar, more specifically known as the 2' carbon. Ribonucleotides, or sorry, deoxyribonucleotides are that molecule without that OH group. A ribonucleotide has that OH group. Hence why we say deoxy, because we lost an oxygen. That's the only difference between the sugars that are found in DNA and RNA molecules. If we go through here, there's this annotation that I wanted you guys to know about. Now, if we look at something like this, P, A, T, C, lowercase p. What does that lowercase p stand for? Go ahead and add to the chat somebody. What does that lowercase p stand for? I'll give you a little bit of a hint. Boss. Yeah, boss. That's what I'll leave as the hint. Phosphate. Perfect, Alondra. Great job. So what this lowercase p indicates on the left-hand side of my molecule is it indicates something about the directionality. That lowercase p there indicates that this molecule, and thinking about the direction that we read a molecule, a DNA molecule, we read in the 5' to the 3' end. So this first lowercase p is the 5' phosphate. This lowercase p right here is the 3' phosphate. So if I showed you this molecule right here, this is phosphorylated. That's the term. This has a phosphate on both the 5' and the 3' end. Now, if I showed you something like this, well, this only has a phosphate on the 5' end. If I showed you this, well, this has neither a 5' phosphate nor a 3' phosphate. So when we talk about DNA molecules, we look at that notation. Now, when it comes to these lowercase, or sorry, the letters that make up a DNA molecule, those are going to be important because what those are those are known as our nitrogenous bases. Nitrogenous bases are one of the components of a nucleotide. This word or these terms are important for you to keep track of. A nucleoside versus a nucleotide. And the way that I've kind of kept this straight over the years is whenever I hear nucleotide, I emphasize, like in my head, I emphasize that T. And the reason that I do that is because, well, a phosphate. I emphasize that T. So a nucleotide has a phosphate. It's helpful for me. I know that's kind of dramatic, but there we go. That's compared to a nucleoside, which does not have a phosphate. So there we go. So this molecule that's shown on the left-hand side or on the right-hand side of our screen is showing the building blocks: a nitrogenous base, a phosphate, and a sugar. So if I were to give you this molecule and say, what did I just highlight? A nucleotide or a nucleoside? Your answer would be a nucleotide. What if I gave you this? What have I highlighted? Answer in the chat what I've highlighted there. Is it a nucleotide or a nucleoside, what I've highlighted? Well, this is the nitrogenous base and the sugar. This is perfect. All right, so that's a nucleoside. So understanding and knowing those distinctions is really important. Now, when we go from there, we also talk about different nucleotides. For instance, one that most people are familiar with is this guy right here, or this guy right here, ATP. People are familiar with that because maybe they took a biology class at some point and you learned about the mitochondria, the powerhouse of the cell, where ATP is generated. You maybe have also kind of heard about ATP as kind of the currency for a cell. That's your energy source. ATP or adenosine triphosphate is a nucleotide. It's a high energy compound, and that is basically how chemical reactions are carried out in a cell. Our our nitrogenous bases fall into two different major categories. Here are the nitrogenous bases, the big groups. They're either pyrimidines or purines. Now, purines are highlighted by two ring structures, whereas pyrimidines are highlighted by a single ring structure. This two-ringed molecule is adenine, and adenine is an example of a purine. This two-ring structure, guanine, is an example also of a purine, but this specific one is guanine. All three of these are pyrimidines, and they are cytosine, thymine, and uracil. I what these nitrogenous bases do is they will make within a DNA double helix, they'll form, and there we are. This is what I was getting at. They'll form hydrogen bonds with one another. That right there is an example of a hydrogen bond. That right there is a hydrogen bond. So between A and T, how many hydrogen bonds do we have? Pretty straightforward there. Between A and T, we have a total of two hydrogen bonds. Meanwhile, between G and C, how many hydrogen bonds do we have between G and C? Well, one, two, and three. We've got three hydrogen bonds between three hydrogen bonds between G and C, two hydrogen bonds between A and T. Now, my question for you guys, is, which do you think are more strongly held together? A and T or G and C? Think about this as power in numbers. What we know is hydrogen bonds aren't super strong, but which ones are more strongly held together? Well, what we're looking for there is G and C. Those are more tightly held together because of the fact that we've got three hydrogen bonds as opposed to two hydrogen bonds. And it's just as simple as the greater number of hydrogen bonds, the more strongly those nitrogenous bases are held together. Okay, so that gets us through there. Now, we're going to talk about lipids. Now, lipids and fats, big umbrella terms there. Lipids, there's a total of 5 different major lipids. We talked mostly about fatty acids. There are 2 major classes of fatty acids, saturated and unsaturated fatty acids. Saturated fatty acids have no double bonds. Unsaturated fatty acids have at least one double bond. Here, what we have in linoleic acid is a molecule with 2 double bonds within our hydrocarbon side chain. So from here on, we've got two double bonds. And I say that just because arguably this double bond we don't really care that much about. So this molecule has two double bonds that we're interested in. That makes this a poly, so multiple, unsaturated fatty acid, a polyunsaturated fatty acid. Alpha linolenic acid is also a polyunsaturated fatty acid. It's got three double bonds. Okay. So with respect to that, one of the things that we talked about that's worth thinking about is the packing of fatty acids. Typically if somebody leads kind of a sedentary lifestyle, they they're not very active in terms of running or any sort of physical activity. Um, it's generally, I think that it's pretty well received that um, they should not have a diet that's high or rich in saturated fatty acids. Instead, they should get more unsaturated fatty acids. And the reason for that is your saturated fatty acids, what they do is they pack really tightly. They pack really well together. And when they pack really well together, that means you can have a high density of fatty acids in kind of a small space. And what that means is you have a compound or a molecule that has arguably a huge amount of energy. And so you have this huge amount of energy compacted in this small area. And if somebody's not very active, well, then that's a lot of energy that's going unused. So that's why it's suggested for lots of people that, based on your lifestyle, you should eat and consume fatty acids accordingly. If you have a very active lifestyle, if you're like a CrossFitter or something like that, then you probably need to ingest more saturated fatty acids because they can pack really well together. And then you have like a concentrated and efficient energy source. In addition to that, we talked about fatty acids and how their physical property of their melting point changed. As the number of carbons increased, so too did their melting point. So if you had a molecule that was 30 carbons versus 12 carbons, well, one of them is going to have a melting point that's very high compared to the smaller one. The smaller the molecule, the fewer carbons, the lower the melting point. Now, we also looked at how that was impacted by, or how melting points were impacted by double bonds and the introduction of double bonds. Well, as you introduce a double bond, that brings your melting point down. So here, what we referenced in particular was we looked at these three. Actually, we could take it beyond that. I'll look at these three. Here we have 18:1. That's an 18 carbon long fatty acid with one double bond. 18:2. That is an 18 carbon long fatty acid with two double bonds. 18:3 is an 18 carbon long fatty acid with three double bonds. The melting points of those, sorry to yawn, the 18:1. So the 18 carbon long fatty acid with one double bond has a melting point of 13.2 degrees. The 18 carbon long fatty acid with no double bonds has a melting point of 69.1 degrees. Okay, so when you introduce one double bond, it takes your melting point from 69.1 degrees Celsius down to 13.2. When you introduce a second double bond, it takes that melting point down to -9. And then a third double bond brings it all the way down to -17 degrees Celsius. Okay, so these are all factors whenever it comes to fatty acids and basically their function and their physical properties. Now, in addition to that, we talked about this process right here, saponification. Saponification is an industrial process by which triacylglycerols are broken down, and this is the process by which soap is made, or soap has historically been made. If we go on from here, another example of a lipid is cholesterol. Cholesterol is a steroid. It is a molecule that's made up of these four big rings down here, as well as this side chain. Now, cholesterol as a steroid is the source of a number of different sex hormones. In addition to that, they can be used for synthesizing like anabolic steroids. Lipid soluble vitamins are another example of well, lipids. For instance, A, D, E, and K are your lipid soluble vitamins. They do a number of different things. Vitamin D is probably the lipid that I think most people are most familiar with, especially in Central Texas, where you can say, hey, look, I've got an infinite supply of vitamin D. And the reason that you can say that is because, well, this molecule right here, 7-dehydrocholesterol, is more or less activated by UV light. So just by getting sun exposure, your body begins to metabolize this molecule into the active forms of vitamin D, which are this guy right here and this guy right here. Now, there are some steps that are required within the liver and the kidney for this to take place, but that's kind of where it gets started. Now, last but not least was our protein ligand complex. We talked about hemoglobin. Now, proteins and ligands, they bind in a non-catalytic fashion. We talked about deoxy and oxyhemoglobin. Oxyhemoglobin is your arterial hemoglobin. That's going to make your red blood cells in your blood red. Your venous hemoglobin has your deoxygenated hemoglobin. And that's what's going to make your venous blood dark purple. So, I apologize. When your blood doesn't have an oxygen concentration, or basically a safe or a healthy oxygen concentration, your blood's going to reflect that and be kind of blue, maybe even a little bit purple. Now, we talked about, with respect to this, hemoglobin and myoglobin. These are two proteins that carry oxygen from your lungs to your peripheral tissues. They do it with a molecule called a prosthetic group. And this specific prosthetic group is heme. Heme is a prosthetic group that, well, basically helps hemoglobin and myoglobin bind to oxygen. So without that prosthetic group, they would not be able to do that. Myoglobin is a protein that's 90% alpha helix, and it's going to, help transport oxygen. Hemoglobin is a protein that transitions between a T or inactive state and an R or an active state. So this is what hemoglobin does. It will be either inactive or active. It's kind of a light switch. Now, hemoglobin is a protein that can bind 4 O2 molecules because each one of the subunits of hemoglobin binds one heme molecule. And that heme brings into it, at its center, an iron ion. And that iron ion helps hemoglobin and myoglobin bind oxygen. Now, what I was or your big takeaways for all of this, I want you to understand that hemoglobin and myoglobin are distinct proteins. That hemoglobin is made up of 4 different subunits. It's not made up of 4 myoglobin monomers. Now, next up, I want you to understand this right here. This is an oxygen binding curve. And what this tells you is this is going to tell you the amount of oxygen that's required to get a protein to be 50% saturated. And what this shows is that this is what is known as a P50. Now, different proteins have different P50s, but this is an important general rule. A high P50 means that your protein has a low affinity for oxygen. A low P50, by contrast, means that your protein has a high affinity for oxygen. So if you're thinking about, well, what's healthy? What would be good? Well, you want hemoglobin that's going to suck up oxygen. It's going to get as much oxygen as it can very easily. So you want hemoglobin within your body to have a low P50 because that means that it can basically, I always kind of think about it as it can find oxygen as the needle in the haystack. It can look at the haystack and be like, there it is. Got it. You want it to have that high affinity. Now, when we go on from here, and this is kind of the I would say one of the most important takeaways that I want you to get is that hemoglobin is a protein that exhibits what's known as positive cooperativity. What that means is that hemoglobin's I guess, first O2 binding is the one that it wants to do the least. So what that means is that the deoxygenated form of hemoglobin wants oxygen less than the fully oxygenated form of hemoglobin. I gave the analogy the other day of, a junior high school dance or something like that. Any sort of, well, I would classify it as any dancing setting where nobody wants to dance, but nobody wants to be the first person to dance. But then being the like 55th person to dance is like, not only do you want to, but you feel kind of obligated to. This is very similar. The last oxygen to bind to hemoglobin is the one that does so most easily. Okay, so I kind of breezed through this, but I gave you guys the practice exam. And I think that, for what it's worth, that practice exam is maybe the most important resource or reference. Um, the practice exam is just like your actual exam in terms of like the number of questions. It has 25 questions. The practice exam isn't worth any points. I can look at this and I can see that we've got one person who took it. Everyone else, I encourage you to work through the same thing. I see that the one person who took it, they went from a 60 to a 100 over those three attempts. I'm not going to say that person's name or anything like that. But just so that person knows, they have 996 more attempts at this exam. Um, when you're taking that practice exam, what I strongly encourage you to do is look at each of the questions and think about it and say, okay, if I change this question, how could I change it where it's kind of getting after the same topic, um, but I'm making it a new question? Think of each one of these questions on this exam as kind of like a learning objective. Um, that's what I would say is the most important thing about this practice exam. Make sure that you're like taking it and taking it critically. So the first question, for instance, is about the old stuff. And that first question says, what is the one letter code of the amino acid arginine? Okay, so you should probably think, okay, well, this is probably asking me a question or on the actual exam. I should probably anticipate a question about a one letter code of an amino acid. The next question is, what is the three-letter code of the amino acid aspartic acid? So then on the actual exam, again, you should probably expect a question about the three-letter code of an amino acid. The third question is, which amino acid is classified as a polar negative residue? We classified amino acids based on, are they non-polar, polar negative, polar positive, or polar uncharged? So you should probably think about, okay, well, maybe he's going to ask a question about a polar positive residue. Um, so anticipate those sorts of questions. Um, what are the chances that I ask a question on the practice exam or the exact same question on the actual exam and the practice exam? Pretty slim. So I wouldn't commit them to memory, but instead I would look at them and be like, okay, I've got to get this so I have basically the same question in mind, or a similar question in mind. Um, I've got to move this down. Okay. But that's what I think is the most valuable thing for you to do in order to prepare for this exam. Take that practice exam as many times as you want. Right now, I'm moving it into the ungraded section of the class, because it's not worth any points. It's not an actual graded assignment. But with that, I'd like to open the floor for any questions. Does anyone have any questions for regarding this upcoming exam or or grading or anything like that. I'm looking over here just because I'm kind of reorganizing parts of our grade book. But if anyone has any questions, please just add it to the chat or you can unmute yourself and ask. Okay, well, if there are no questions, I'm going to go ahead and stop.